So welcome everybody to Strait Science. Strait Science is a evening science presentation series put on by UAF, University of Alaska Fairbanks Northwest Campus, and in Nome, and UAF Alaska Sea Grant in Nome. And you're in the home office tonight. We also acknowledge the indigenous peoples upon whose customary lands our UAF Alaska Sea Grant and Northwest Campus offices reside. In the Bering Strait region, our campus for UAF is located on the customary lands of the Inupiaq, Siberian Yupik, and Yupik peoples. And tonight's speaker is a senior scientist in the biology department at Woods Hole Oceanographic Institution. And he runs a very active and very comprehensive program on harmful algal blooms. And his current research focus is on harmful algal blooms in the Arctic. And tonight, Don is going to give his research results and tell us a little bit first about harmful algal blooms, and then go into what he's found and some of the potential issues with that. We hope to hear people at the end of his presentation. He's asked if you could just hold off your questions, unless they're super burning questions. Hold off your questions till the end. And also know that he is in, where are you, Don? Where are you located?
Massachusetts.
No, right now.
In my office, you mean? Or
yeah, aren't you in Mexico?
No. That's a virtual conference.
Okay, so all right, my misunderstanding. So he's been in a virtual conference doing Zoom all day. It is 10:30 at night. He's been at this Zoom business all day, and he is taking his time because it's that important to talk to the region. And we really appreciate it, Don. So if you drift off, we'll somehow call you or buzz you. But we really appreciate the fact that you're doing this at such a late hour. So
Great.
Again, thanks everybody for coming. Callers will have the preference when it comes to questions or any kind of interruptions, because it's really hard to be a caller on a Zoom call. So I will be checking that out. Last but not least, there's also, for those of you who may be new to Zoom, there is at the bottom of your screen a little cartoon bubble that says chat. Looks like a cartoon bubble. And if you click on that, you'll get a screen, and you'll be able to text or type in a message either to an individual or to everyone in the group. So you can also ask your questions. If you don't want to voice up, you can, sorry callers, that won't be an option for you. You will have to voice up. But for everyone on the computer, you can either voice up, or if you don't feel comfortable with that, you can write in your message. And I'll be monitoring the chat. So that should be it. Thank you so much. Take it away, Don Anderson, and let's hear what's coming at us for our Bering and Chukchi Seas regarding harmful algal blooms. Thank you.
All right, great. Well, thank you, Gay, and hello to everyone. I'm going to try to tell you something about some relatively recent research results that are scientifically interesting, but also very important from a social viewpoint as well, something that's very important to your region. And as you see on the title slide here, this is not just me. There's a large group of people who have been part of this project, and I'm very grateful to all of that support. And what we're going to talk about are what some of you may have heard the term red tide. Gay and I, we use harmful algal bloom, or HAB, and I'll try to explain to you what those are, and then what we're seeing up in the Alaskan Arctic these days. But to start off, and to make sure we all understand what HABs are, I'm actually going to start with a picture on land, and it's a pretty picture, and you might look at that and see just a bunch of flowers. But if you were a botanist, you would know that there's a lot going on there in that picture. That some of these plants, well, there's many of them that are blooming, as we would call it. They're proliferating, but they're also fighting for space, and some of them may be overgrowing other ones. Some could well be producing chemicals that try to repel the other and keep the others from growing. Some are producing chemicals that would keep animals from eating them. Some are growing fast, some are growing slow. And if you looked at this in a month or two, there might be all these different flowers growing there. Well, that is something that you're sort of familiar with. This is the same thing. I look at the ocean the same way you might look at the land. These are tiny plants in the ocean. Just under the microscope, you see many, many of them here. There are different species. These golden ones are the ones we're going to talk about tonight because they produce toxins that can be very dangerous. But there's other algae in here. These are microscopic algae that are perfectly harmless and beneficial, and they produce a lot of the oxygen we breathe, and they're the base of the marine food chain. So the ocean's filled with thousands and thousands of species of algae, and there's a few hundred that are dangerous or harmful in different ways. And these plants, these microscopic plants, when they grow, they don't get big like a tree does. They become more numerous. So one cell becomes two, and the two grow and divide and become four, and you keep going that way, and you can actually get to the point where there's enough of them to change the color of the water. And here's what we call a bloom of algae. In this case, this tiny little area in Norway. Here's one that's bigger. You can see this bloom here. This is in the Potomac River. And then some of them are absolutely massive in scale. Here's one you can see from space, stretching from Korea almost over to Japan. Now, if we look more closely at what these types of blooms we could have, here's one. This is not a picture it's doctored or photoshopped. This is real. It looks like blood or tomato soup. It's an organism called Noctiluca, which I imagine some of you have seen up there in Alaska, but it's non-toxic. In a sense, it's harmless in this type of configuration where the bloom's in fairly deep water. But when that bloom or others like it, these non-toxic blooms that have high biomass, in other words, large numbers of cells present, when that moves into shallower waters and starts to decay and run out of its nutrients and to die off, then that biomass gets consumed by bacteria that use up oxygen, and you have this situation where everything in the bottom dies because of a lack of oxygen, not because of toxins, but just because of a lack of oxygen. And that's another type of harmful algal bloom. And you might see a scene like this. This was from Chile, where they had a huge outbreak like that of anoxia, or lack of oxygen, that just killed shellfish by the millions and billions. And you've had some of these events. There was one down near Homer in Kachemak Bay that was called a beer tide because of its color, but it caused a lot of oxygen problems. But then there's these types of blooms, and this one, the water's brownish and it looks discolored because of the number of algae that are present. And all of these dead fish, though, in this case, they have been killed by toxins that are produced by those algae that have actually directly killed the fish. This is not an oxygen problem. And not only these little tiny fish, but you can see these monster Goliath grouper. You can see other fish dead by the millions along beaches. So in this case, there's a toxin that can actually kill fish. And this particular toxin doesn't occur up in your waters yet, but there are other fish killing type species that might occur in your area. But this is really what we're going to talk about tonight. One of these are human poisoning syndromes that happen when we eat toxic shellfish. Shellfish are filter feeders, meaning that they filter the water, the seawater through them. A little mussel like this can actually filter about 10 liters or 10 quarts of water in an hour and take all of the algae that's in there out of there as food. And because they're so good at doing that, they accumulate large amounts of the toxins that might be in the algae that they're eating. And they're processing so much water that they become very dangerous. And that's one of the reasons that shellfish are an important vector, we call it, for the toxin. And so shellfish then when they filter different types of algae from the water, accumulate different toxins, which then cause different symptoms in humans who eat them. And we have all these acronyms: PSP, NSP, ASP, and so forth. And you can see they're based on some of the symptomology. The one we'll talk about, paralytic shellfish poisoning, is a paralyzing toxin. It can keep you from breathing, keep your muscles from moving. This one, well, let me just say that the ones that you need to start worrying about in the Arctic up there are PSP and ASP. This one I'm not going to talk about after this moment, but it's worth its own straight talk seminar at some point, because this toxin not only causes wildlife mortalities and so forth, but in humans that get poisoned, it can actually cause permanent memory loss, short-term memory loss, just from eating some mussels like this. But the focus now is going to be on paralytic shellfish poisoning or PSP. So if we look at the world, and each one of these dots shows where PSP has occurred at a regulatory level, where it's dangerous. And you can see it is the most widespread of all of these HAB poisoning syndromes that we have. And when I see this map, my eye often goes up, and it used to especially go up here above Alaska, or above North America, or even above Russia and Asia, and notice there's just none of these red circles. And is that because the problem isn't there, or is it because there are no observations? And so for years I was trying to get funding to go up and study these northern areas, and was able to get some money to go up here on the coast of Greenland, but kept getting declined to work up here. But I can say that finally the funding has come in, and we are actually now starting to put some dots up here. We now have this problem up into this area. Now, again, a little more background. This is the organism we're talking about tonight. It's called Alexandrium catenella. You see two cells here that swim. They're about the size of the sharpest point of a pin, just barely visible to the naked eye. And this also, though, is that same organism, but it's a different life cycle stage or life history stage. And this is called a cyst. And think of it as a seed in a higher plant. It's a resting stage. It's a dormant stage. And you can see how it fits into the life history here. These cysts are there in the sediments, and in your region, they're in the sediments for most of the year, 9-10 months of the year. They're just down there dormant. And then they germinate and they hatch. And I'll explain a little more about what drives that. And they produce what we call vegetative cells. And just think of it as a swimming cell, an active cell that catches the sun's energy and photosynthesizes, divides like I described earlier, to make all these cells that cause a bloom, produce a lot of toxin. Then at some point, this bloom starts to run out of the nutrients that fuel it, and they make gametes that fuse together, make the zygote, and that becomes the cyst again. And so that's an important factor for you to remember as we talk tonight about what's going on. Now, let's take a look, though, at the way this toxin affects different levels of the food chain. Up here, you see the Alexandrium cells, as if you're looking at them under a microscope. And in many of these other parts of the world, including where I've studied them a lot in the Northeast, we have really been focusing on this part of this diagram. And especially here, where the shellfish, the mussels, the oysters, the scallops are filtering the water, accumulating the toxin from these cells, and then being dangerous to the consumers, the humans and other animals that might directly eat the shellfish. But we also know that these cells are eaten by the zooplankton, the little tiny swimming animals that like krill, that will eat them and also retain the toxin. And these are in turn eaten by forage fish like herring or menhaden or capelin or sand lance, and which are in turn then eaten by predatory fish and even these top predators. And most of our work, or not just me, but many of my colleagues, has been over here. And it's as these organisms are now showing up in these environments, like now above Alaska, a lot of this aspect of the food web is coming into play, and we're finding how much we don't know about the way these toxins move. And I'll talk more about that in a second. And just so you realize how serious this can be in terms of that food web transfer. This is a paper that I'm part of that was written back in 1989, so it's a long time ago, but it was an extraordinary event where here in Massachusetts, in 1 month, 14 humpback whales died in that 1 month period. An amazing event. There were people out whale watching, and these whales would be breaching, and everyone's all excited. And an hour later, that some of those same whales were just floating and dead. It was people thought there was someone with poison, someone with high-powered rifles that was killing them. Who knows? But it turned out, as we published, that it was this toxin that I'm talking about, paralytic shellfish toxins, that killed those whales. So more than 50 years worth of mortality in 1 month by that toxin. And I wish I had time. If you could read this, it says, we propose a line of evidence to explain how whales, by virtue of their diving adaptation, may be particularly vulnerable to this systemic, hard to read it myself, neurotoxin. So maybe someone can ask that as a question, but right now I won't have time if I go into it. But it's a fascinating story about whales and their diving physiology and how it makes them vulnerable. All right, so now let's take a different map and look at the United States and all the different types of HAB problems that we have. And I show this for 2 reasons. One is to highlight that here in Alaska, the problem with PSP, this red coloration, is we've known about it for a long time, for hundreds of years, really, in the southeast and in the south central Alaska. The problem has been there for a long time, but we haven't had much knowledge or experience with it up further. And I'll talk about that. But I also want to highlight where I have done a lot of work over the years here in what we call the Gulf of Maine. And it stretches from the state of Maine and Canadian border down into Massachusetts. And the reason I'm showing you this is that it provides a very good context for what we're observing in your waters, because we understand what's going on there and we've studied it for a long time. So I'm going to spend a little bit of time telling you what happens there. So here's the Gulf of Maine, and notice these big arrows. These are currents, a big coastal current that we call the Maine coastal current that branches and comes down here. It's got a lot of other subcurrents, but it's this big current that moves along the coast from north to south. And I told you about the cysts, or these seeds for Alexandrium, and we've mapped them out up there. And here you see in red the highest concentrations, and these are what we would call seed beds or cyst beds. They're just accumulation zones in the mud of these cysts. And and I will point out, by the way, that these numbers go up to about 1,000 or 2,000, the units that we use there. And you see 2 seed beds in this region. And now this is sort of, I'll try to talk you through what happens. Here's one of those cyst seed beds, here's another, and the coastal current moves down here. And I've got a little simulation model going over here from a computer that shows you what is basically happening. The cysts are germinating, they're moving down in this coastal current, heading from north to south. They come out here around George's Bank. These are basically very seasonal events. They're annually recurrent because the cysts that are germinating form blooms that deposit new cysts that started again the following year. In southern waters, like down here, the problem is being carried to them. There aren't no cysts down here to speak of in any large numbers. So the problem is introduced from the north. And there's considerable interannual variability in the number of cysts, but the location of these cyst seed beds is persistent. And I'm telling you this because this very same picture now applies in your region. So let's take a look at your currents. Here you've got a current from the eastern part of the Bering Sea and the Anadyr from here on the west along the coast of Russia, and they hit the Bering Strait, and they sort of form these water masses and branches. This central one, the central channel one, has got different names. It's sometimes called Pacific Summer Water or Bering Summer Water. And this one, though, is the Alaskan Coastal Current, or the Alaskan Coastal Water, is one that we're focusing on a lot, which comes out here into Barrow Canyon and towards the Beaufort Sea. So those, you have now this current system, in this case, flowing from south to north. And I'll come back to this image in a bit. So again, by way of background, we, as I told you, this whole region in the southeast and south central, we've known had PSP problems and for a long time. But in 2016, one of my colleagues, Kathy Lefebvre, produced this really landmark paper. And what this shows, they analyzed about 900 marine mammals from this entire region, both harvested and stranded, 13 different species. And the ones that are colored in sort of maroon or reddish are ones that have that ASP, that amnesic shellfish poisoning toxin, the one that has the memory loss associated with it. And the blue ones are the saxitoxin, the PSP toxin. And what this shows you, and it was no surprise that animals down here contain both of those toxins, but look up here in the northern Bering Sea and in the Strait, and even up here in the Chukchi, and as you go into the Beaufort, the toxins there. So here's some clear indication that the organisms that cause these problems and their toxins are there. And then while I was still trying to get funding to work up here, some Japanese colleagues did get funded to work here and published some very important papers about Alexandrium. And on the left, what you see here are about 8 or 9 stations that they sampled and counted the cysts, those seeds in the sediments. And what you see is this huge circle here near Ledyard Bay, which is roughly 10,000 cysts per cubic centimeter. Remember what I showed you in the Gulf of Maine, those numbers were about 1,000 or maximum 2,000. This one says over 10,000 there. And I remember seeing this paper and thinking, my gosh, that's a huge number of cysts there. And then in a companion paper, they have, this is a little harder to see because it's small, but these white circles, now they're looking in the water column at those vegetative swimming cells. This is the sediments, this is up on the surface. And these are cruises that are, some roughly a month apart. And what you're seeing here is a population moving right through Bering Strait, being carried from these southern waters, let's say the northern Bering Sea or western Bering Sea or wherever, through the Strait and into this region, the Chukchi region. And then the same thing a month later, and then the same thing here. And this paper basically said that the cyst.
Don,
Sure.
I'm going to interrupt just for 1 sec. For those that are following along on a presentation that's been emailed, so he's on a black and white slide with 3 black and white maps on the right, and the area for the callers that may not have a presentation in front of them, your cursor was kind of for the western Bering. You were really in the Gulf of Anadyr, if that helps our region.
Good, thank you.
Thank
yeah,
you.
I keep forgetting there are callers who aren't going to have this in front of them. I'll try to be better going forward. So the conclusion of this very important Japanese paper, though, was that the problems up in the Chukchi and the area north of the Strait were being carried there from the south through this, what they call Pacific Summer Water. And when I heard that and saw all the large number of cysts, I remember thinking, why are there so many cysts there? And that's where there was a great paper written not about algae, but about zooplankton, these little animals. And it was called The Trails of Life and Death by Paul Wassman. And the idea was that there are animals like these zooplankton that get washed into the Arctic where the waters are very cold. And because it's so cold, their physiology slows down and they are not able to go through the entire life cycle that they usually go through, laying eggs that hatch and produce these naupliar stages that grow and eventually give you to an adult. So they call this transport pathway the trail of death. And I thought, well, we can hypothesize that Alexandrium is doing the same thing, that it's being carried from the south in these relatively warm surface waters, and that it would form cysts up there in that region, in the Chukchi region, that fall to bottom sediments where the temperatures would be too cold to support significant germination. And so what you'd have is a situation where you get this continued input but very few losses from germination or hatching of these cysts. And you therefore would theoretically accumulate to these really high numbers that were being reported there. And it's worth pointing out that these cysts can survive anywhere from decades to even a century. So what this would represent, and this is one of the proposals I wrote, and I called that cyst seedbed a sleeping giant. It's something that's waiting to wake up. And when it does, it's so many numbers and it's so huge that it could really be a significant event for the region. So that's one explanation for why there are so many blooms. So with some good fortune in the proposal writing, we have ultimately been able to get funding from a number of different sources now, and we have been able to benefit from a lot of the cruises that happen in the region, starting typically around Nome and St. Lawrence and going all the way up to Point Barrow, Ukqiavik, and the entrance to the Beaufort. And we've been on the Healy and a number of other vessels collecting water samples as well as sediment samples. And so now let's go back and take a look at the cysts. And so this figure shows a map of the cyst distribution of Alexandrium catenella in the region, stretching from, let's say, south of St. Lawrence up into the beginning of the Beaufort Sea. And for those on the phone, what you would see is a big red bullseye somewhere around Ledyard Bay, grading to sort of a light yellow color, meaning fewer cysts, but still good numbers as you head towards the Strait, lower numbers in the Strait and much lower numbers further south in the northern part of the Bering Sea. And as you go up to the north and east, the numbers sort of drop off as you head towards the icy cape and Barrow Canyon. But then we find a second cyst seedbed, a second bullseye, if you will, right near Point Barrow. And basically, this is a map that we got by combining two different cruises from 2 different years. And what it shows is first of all, a massive cyst distribution. It's huge, and I'll tell you how huge in a second. And it's persistent because we know that 10 years early, the Japanese were here in that same region finding large numbers of cysts near Ledyard Bay, and we're seeing the same thing now and have multiple years in a row. So it stretches at least 200 kilometers offshore. We can't go any further because that's Russian waters. So if you could see the map, there'd be a straight line where we just can't any more samples. But our samples right up to the edge of that line are high concentration. So we know this feature extends further into Russian waters and is even bigger than is shown here. And it stretches a good 600 or kilometers or more along shore. And the concentrations we're seeing are as high as 17,000 cysts per cubic centimeter. So even higher than in the Japanese paper and much, much, much higher than we've seen in other areas. And we see positive but low numbers elsewhere in the Beaufort area and in the northern Bering Sea. And then this the second seedbed that I mentioned is near near Barrow Canyon, also has very, very high cyst abundance. So what I've got here is a slide where I've got on the map of the cyst, let's call it the Alaskan Arctic cyst distribution. And on the right is the one in the Gulf of Maine. And these are drawn on exactly the same scale, an aerial scale. And the color bar is also exactly the same. And what you can see is that the Gulf of Maine one has far fewer cysts and is much, much smaller. And to put that in context, this Alaskan Arctic cyst seedbed is 6 times larger in area than that equivalent feature in the Gulf of Maine. And it contains roughly 15 times more cysts. But again, for context, that Gulf of Maine cyst seedbed, as sort of wimpy as it looks right now, it looks just small, has supported large scale, meaning going from Canada down to Massachusetts, annually recurrent Alexandrium blooms for decades, year after year after year after year. And the one you've got out here is much bigger and much more dense. All right. Now, let's take a look. We're moving now up into the water surface. And these are the vegetative cells. And we have 2 years worth of data that I'm showing. Unfortunately, we'd have much more except for COVID, stopping and canceling a lot of cruises. And basically what we see in both 2018 and 2019 are dense blooms in the surface waters in the basically July, August, early September timeframes. Both years, there were dangerously high cell densities that covered large areas, but they were distributed differently. And there's some real good information for us about why the dynamics of these blooms from that different distribution. So let's go through this one by one. In 2018, there is a map that shows, again, another bullseye of reds and oranges, meaning high cell densities in the surface water, sitting directly over that cyst seedbed around Ledyard Bay, with lower cell densities south and east. And again, we think this feature was much larger, but we couldn't go into these Russian waters. And this is important. On the right is a diagram, what we call a TS diagram. It's a plot of temperature versus salinity. And it's a way of looking at, in a sense, the fingerprint of water masses, because certain water masses have salinities and temperatures that are characteristic. And you can measure those. And as you move through the water masses or move to different areas, you look for those fingerprints for the different water masses. And so you can see this ACW, that stands for the Alaskan coastal water, and it's warmer and it's fresher. And then there's BSW, this Bering summer water, that's a little saltier. And if you then say with this color, you say what water mass were these cells associated with, you can see it falls into that fingerprint of the Alaskan coastal water, that near shore current that you see and that you saw on the map of the currents, the one that runs along through the Strait and near the Point Hope and Icy Cape and out towards Barrow, right near shore. So the cells are associated with that near shore current, and the cell abundances were roughly 5,000 cells per liter, which is really an order of magnitude more than the levels that we know from Southeast Alaska can be really dangerous, both to humans and to wildlife. So it's a big bloom and it's quite dense. All right, now I'm going to look at 2019, and there we had a great opportunity because there were 2 cruises that we got samples from that were separated by about a month, a little less, but about a month. And the first one shows, in this case, the bullseye where the most cells are, this big patch of cells that would represent a bloom, is near Kotzebue Sound. It's just north of Bering Strait. And everything around it is fairly blue, dark blue, meaning there aren't very many cells there. And if we look at that TS diagram, now you see that those cells are in this BSW, the Bering summer water, just like the Japanese had seen in their study. This is water coming through the strait from the south, BSW. And then, though, take a look several weeks later at the second cruise. Now we see the yellows and the oranges up northeast of Ledyard Bay towards Point Barrow. And if you take a look at this TS diagram at that point, the cells are associated with this Alaskan coastal water. And in this case, what we can't see very well is that there's another patch of cells that we picked up right to the slightly to the east of Utqiagvik, right over that other cyst seedbed as well. So what does this mean? Well, remember these coastal currents. This is the Bering summer water in the middle. This is the Alaskan coastal water along the coast. What we've got is basically a situation where the first bloom we saw was one being carried into the region, whereas the second bloom is one that we believe was germinating from that cyst seedbed. And again, the evidence is because it's associated with a different water mass, but also we did calculations with current data that was from the ships and other data, and it would just take too long for the bloom picked up in the first cruise to travel up to where it was observed, in the second cruise. It was basically, it can't get there that fast. And so we believe, go ahead.
Don, if I can just cut in. So I think this is an important slide, and this is labeled 2019 Vegetative Cell Distribution and Dynamics at the top. And the areas that, what you were calling, I think, a bullseye in the top slide on the left, is an area that's just southeast of Point Hope. Maybe 50 miles or so southeast of Point Hope. That's a very high concentration is what he was talking about. And the second slide where it had traveled, I think, Don, too fast, up, so it had to grow, was an area in Ledyard Bay. So basically north of Utqiagvik, right on down to the nearshore waters and down south to sort of Point Lay and sort of between Point Lay and Barrow, including Wainwright. Thank you very much. Sorry.
Great, thank you for that geographic clarification. I'm trying, but I don't know my geography as well as you do, that's for sure. But again, look at these TS diagrams, and you can just see that these blooms are in different water masses, and that's just so important. All right, so let's talk now about some temperature data and how they help us interpret what's going on. So here is a plot that shows two lines, one that's for Ledyard Bay and one that's for the Barrow area. And there are basically four data points in each line, and each one represents a 5-year interval. So this goes back 20 years. So 1999 to 2003 is the first one. The last one's 2014 to 2018. And it's just temperature on the y-axis. And what we have here then is the average, the bottom water temperatures during the summer, July and August, September, in the Ledyard Bay and the Barrow areas. And what you can see from this is that over the last decade or two, the water temperature has gone up roughly 2 degrees centigrade in those areas. And so, all right, what does that mean? Well, let's be a biologist here that studied Alexandrium. And what we have in this diagram, it is a curve that is showing the days until germination on the y-axis and temperature on the x-axis. And what we've done is taken mud from a number of different areas on Cape Cod and the Gulf of Maine, up in the Chukchi Sea, and germinated or exposed those sediments to different temperatures and measured how long it takes the cysts to germinate. And what happens is all of the data falls on this beautiful curve that you can fit with something that we would call a heating degree day curve. And you probably all hear about that up in Alaska because you want to know how cold is your winter. And that's basically a degree days. It's a metric that incorporates both temperature and time. And to explain it, if I look at this curve, and I say, I'll incubate some cysts at 10 degrees. How long does it take to germinate? It takes 8.5 days. And if I incubate those cysts at 1 degree, it's going to take 85 days. So this red line is 85-degree days. And all of these cysts have this function of they require 85-degree days to germinate. So that's a wonderful fact, because we can now take it. And in this next slide, you see two panels. The top is the one we saw earlier, which was temperature in these 5-year intervals. And down below, we've calculated how many more cysts are actually going to germinate in that summertime bloom period because it's warmer. And what you see is roughly a factor of 2. So we're getting twice as many cysts over the last 10 or 20 years germinating than was the case before. And what does that mean? Well, I know Dean Stockwell's on here. If he were like me, he'd say, well, that's no big deal. It's just one division, and these cells divide many times when they're making a bloom. But it is significant, because in the Gulf of Maine, again, where we've worked on this and where we really have established relationships between cyst abundance and the length of the coastline that is closed every year, a 2-fold increase in this germination, the inoculum for the bloom, would give you 2.3-fold increase in the length of coastline closed to harvesting because it's dangerous, and as much as a 6.5-fold increase in the cumulative amount of toxicity we might measure along that coast. So it's a big deal, but it's not the whole story. This next diagram, which for those of you trying to look at the PDF, it starts with warming temperatures have also expanded the environmental window during which blooms can form, and it shows 4 maps on the right side. And the bottom line for this is that the cysts are going to germinate earlier, start the bloom earlier. It's like out here we have dandelions and crabgrass in April on our lawns, and imagine that they were going to happen in March or February instead. That's what we're seeing. And to explain it, on the top left panel is temperature in the decade from 1999 to 2008, and the colors show you how warm things are throughout your region. And then the right panel at B shows from 2009 to 2018, the temperatures there. You can see it's gotten warmer. It's gotten more oranges and reds. And if you subtract one from the other, you get panel C. And this is the difference in temperature. And that really shows you anywhere from 2 to 4 degrees changes in bottom water temperatures throughout the Ledyard Bay area, right near Point Hope and so forth. It's showing you the colors indicative of that kind of warming. And then on the final panel on the right, you see in green is outlining those 2 cyst seed beds, and the coloring underneath them is just a measure of what's the difference in the number of days until germination. And you can see that there's these darker colors are 20 days, as much as 3 weeks difference in germination because the water is warmer. So what does this all mean? Well, with doubling times, and I'll talk about this in a second, in the surface waters that we've measured, the temperatures of the surface waters, these cells can double. One cell makes 2 in anywhere from 2 to 3 to 4 days at those temperatures. Well, if you give a 20-day head start, think about how many divisions that is that you have been able to get this bloom going because of that head start, if you will. Okay, so we could summarize all this by saying that recent warming that we're seeing in the bottom water supports the earlier and faster germination. It also supports longer periods favorable to blooms. That's because the surface waters are warm, and more rapid cell division and bloom development. So what you've got then is a dramatic increase in the potential for what we would say is local initiation of blooms. In other words, not this transport, but blooms that are starting within that region, inoculated from that region, from those two, Ledyard Bay and the Barrow cyst seedbeds. So this is saying that that sleeping giant has woken up, that the cysts are functioning, the cyst seedbeds are functioning. That trail of death hypothesis may have been valid in the past, who knows, 10, 20, 30 years ago, but it's not valid now. And here's I'm sure some of you or who are on the call recognize this image. You've seen a lot like it. How warm you had, this is 2019 temperatures compared to this 30-year average, and you see surface waters are 3, 4, 5, even 6 degrees warmer in places. In again, centigrade, you want Fahrenheit, roughly double that, and real big changes that you all are experiencing, and I don't need to tell you about it. But what does that mean for our vegetative cells? Well, it helps them grow faster. Here's a figure on the right. It shows that map of the cells that are in the surface water, with the big bullseye sitting off of Ledyard Bay and Point Hope. And on the left, it shows you a plot of, on the y-axis, the growth rate of the Alexandrium cells, and on the bottom, its temperature. And these different curves show you different isolates, some from Iceland, from Greenland, from the Chukchi. The red one is from the Chukchi Sea, and then the Gulf of Maine. They all behave roughly the same, but if you just look at the red one, you can see this blue bar shows you the temperature range that we observed in those surface waters when this map was made on the right, roughly 8 to 10 degrees. And what it says is these cells are growing anywhere between 0.2 and 0.3 divisions per day. And that is roughly 70% of their maximum rate. So the surface waters are more than supportive of growth. Okay, so there you've seen what we've found. Huge cyst seedbed, conditions conducive to germination, conditions in surface waters conducive to growth, 2 different pathways for blooms to get into the region, 2 different mechanisms. So what does it mean now for the people in the communities of your region? And I look at human health issues, look at ecosystem health. I'm going to start here with ecosystem health because that's the way you get to human health issues also. And remember this diagram about all of the different layers of the food web where this toxin can move. And this is where I'm borrowing heavily from work from 2 people, Kathy LaFay and Steve Kibler, both working at NOAA in different locations, different coasts, both working with me as co-PIs on some projects that we're supported on. One of them is listed here as showing the trophic transfer and effects of algal toxins in Arctic food webs. And Kathy's the one that produced this sort of landmark paper on the distribution of toxins in marine mammals all around Alaska. So one of the bottom lines of what we've been doing so far, we've been collecting samples from a lot of these cruises of clams and worms and fish and birds and mammals and so forth, and trying to measure toxins in all these different tissues. And the bottom line is that we don't know yet if toxin levels in Arctic food webs are reaching high enough concentrations to cause health impacts in marine mammals in that region. We know that these toxins can do that in other regions, but we don't yet know if the levels are high enough to cause impacts up there. And there's a number of questions that we are trying to address in these projects. What I'll do just to move on is to show you some data from sampling in 2019. And this shows 3 panels showing on the left, well, it's basically the region stretching from a little south of St. Lawrence, the northern Bering Sea, up to the Chukchi and the beginning of the Beaufort Sea. And on the left, you see the zooplankton that were analyzed. And the colors are showing you the amount of toxin that's in the zooplankton. And the best way to think of it is the red ones are ones where that is over the FDA regulatory limit for this saxitoxin. And the orange would be sort of half of that regulatory limit. Yellow just means it was detectable. and white means there was nothing detected. So what you see in the zooplankton is there is toxin in almost all of the zooplankton sampled. There are a few that are really quite hot, but most of them are what we'd say low. They're low levels, but there are a few that are high. The middle panel shows you clams and worms. Worms are important food for so many animals in the ocean, and they're important vector for these toxins too. And there what we see are basically, again, low to high levels. And notice in the clams, there's at least 3 that were where clams were picked up in these offshore areas that were above regulatory levels. So they would be quarantined. They would not, you should not eat those kind of clams because they're above the action limit for that toxin. And then if you looked at the fish that were sampled in our studies, again, many of these are yellow, a few are red or orange, but very, very low to moderate levels at most in the fish. But the fish bears some more scrutiny. This is work that Steve Kibler has been presenting and is writing a paper on right now. He was great to give me these slides. And the first one shows saxitoxins in the forage fish. But remember, this is Southeast Alaska, an area with big blooms, but persistent blooms that have happened a long time, and not necessarily equivalent to what's happened up in the north. But what you see here are, they had 350 samples from 14 different species of forage fish. You can recognize the names: the capelin, the sand lance, even the juveniles of the coho salmon or pink salmon. This one called Dolly Barden, which I guess is a juvenile char. But this is a log scale, and what it shows is that, and this red line is that 80 microgram action limit. And what you see is that most of the measurements in all of these forage fish are below that 80 microgram threshold, but there are some that are higher. And since this is a log scale, some of them are quite a bit higher, particularly here, the sand lance and the herring, filter feeding fish that are going to accumulate these toxins as they go through a bloom. And so the salmon species and so forth basically had low levels, but detectable levels. And so 60% of the forage fish that they analyzed contained these detectable levels of toxins, but they were at low levels in most cases. But it does indicate that forage fish are likely a substantial source of toxins that will be transferred to marine mammals, seabirds, and other animals through the food web. So let's look now at the predatory fish. And this shows just 6 of them: pink salmon, sockeye salmon, coho, chinook, chum, and halibut. And again, it's a slightly different scale. The red line shows you that regulatory limit. And what you can see is that most of the data are well below that regulatory line. Every once in a while you get a number above it, and those numbers are broken down into different tissue compartments. The digestive organs would be the stomach and so forth, the liver, the kidney, and then the muscle and the roe or the eggs. And what the highest numbers that you're getting in these predatory fish are in the liver and the kidney. And thankfully, if you look at the muscle and the roe, they're all low, well down below this regulatory limit. And on the East Coast, we've studied salmon and cod and other fish as well. And we've generally felt that the fish are sensitive enough to these toxins that they do die before they accumulate levels that are going to be dangerous to people. But notice the liver and the kidney, that these were where we found the highest toxin levels. And so, I want to go back and point out, this was Southeast Alaska. The numbers we've been getting so far up north haven't been this high. So in Arctic waters, we've had very low numbers in fish, but there is useful information in this Southeast and South Central data. In particular, these toxins are relatively common in predatory fish, which makes sense given we saw so much in the forage fish. But the toxin levels in muscle tissues or fillets are very low, and they indicate a very low risk to human health from salmon and halibut consumption. Now, remember, everything I'm saying is a caveat. I'm a scientist. I'm talking about these numbers. I'm not a health official. I'm just trying to communicate to you what we think might be the case, but we're not advising you should or should not eat this or that. Make sure you understand that. So, but it looks like these low levels in this muscle tissue of the fish means things are relatively safe, and there's also little risk to the commercial fishing industry. But given the relatively high levels in those kidney and liver and so forth, there may be some risk to those people who might eat those. I gather that some native Alaskans do consume salmon livers, or that those sort of organs, or sometimes you put an entire fish into a soup. So, all right, so to try to wrap up here, basically our findings raise concerns for increasing risks of these algal toxin exposure in these Arctic food webs. For why? Because we already know that these toxins are out there. Not only our recent measurements, but Kathy's paper in 2016 and other lines of evidence. We know that the ocean is warming, and that's giving you conditions favorable for the bloom development. And these changes are all more favorable for algal, germination and growth. And now I want to just quickly go to the human health things and to try to stay away from any sort of issues that we might interpret me saying do or don't eat that. I want to tell you first of all, tell you about PSP. I haven't described it in real detail, but it is a very serious illness that is caused by eating the shellfish that have accumulated toxins. It is a very potent toxin. It's 1,000 times more potent than cyanide. About 1 milligram can kill an adult. And some of you should know the symptoms. Often one of the earliest signs would be tingling of your lips and your tongue within minutes of eating poisonous shellfish, or maybe an hour or two. And those symptoms may progress where you get tingling in your fingers and your toes, and you can start losing control of your arms and your legs. And you're going to have difficulty breathing because your muscles aren't going to work that drive your diaphragm. And basically you can become paralyzed and you can die in as little as 2 hours because your breathing can become paralyzed. So it's important to know these symptoms. In terms of treatment, the only treatment is something that helps you breathe: a mechanical respirator, an iron lung, and oxygen. So if any of these symptoms are exhibited, you really should call 911 and get some help. And in terms of resources, your Department of Environmental Health has a great website with a lot of details. Here's the URL, and I know Gay has those and is putting that in the chat. But if you just looked up paralytic shellfish poisoning under DEC, you would find this, and it gives you lots of useful information. It says don't eat shellfish harvested from untested Alaskan beaches. Don't eat the viscera or the guts of crabs because PSP toxins can be found there. There's a lot of useful information. Another useful resource is a fact sheet that Gay and me and several others put together when we found those toxic clams offshore in the Ledyard Bay and by the Strait. And that's very useful. That URL is here, and Gay will put that in the chat as well. But I thought I'd highlight just a few things that we wrote in there. And this says thoughts on subsistence uses of marine wildlife. And pardon me if I read some of it, but those who are on the phone can't read. So based on our current understanding of these toxins in many other parts of the world, we think that mussel and blubber are not likely to accumulate saxitoxin in levels that pose a human health hazard. But these tissues have not been fully tested, but there's reasons to believe that they're not going to accumulate toxin. I won't go into those reasons, but they're valid scientific reasons. And thorough cleaning of the inside of marine mammal intestines and stomach contents is a real important aspect of food preparation. We don't know if that's going to totally self-guard against ingesting toxins, but those are the tissues you also want to get rid of if it's a certain season when this bloom might be happening. So eating intestines, stomach, and or their contents in areas with known toxins has the same risk as eating a clam that might have been from the same areas. Okay, but we do also want to emphasize that no one reported PSP symptoms from eating traditional seafoods in Bering Strait or the Chukchi Sea coastal communities during 2018 and 2019 when we saw those blooms that I showed you. So then we're also trying to point out that eating clams, crabs, and other shellfish has always carried a risk of ingesting toxins. But therefore, be aware. I know that one of the delicacies is eating clams from the stomach of a walrus or a seal, but be aware. It can be dangerous. You can't see, smell, or taste the toxins. Cooking or freezing is not going to denature them or change them. They will still be active. So remain vigilant and observant and communicate to a lot of people. And again, on this slide and on this fact sheet are a lot of people that you could talk with, including Gay. And notice that it says, if you see any marine wildlife acting in an unusual manner or dead in the Bering Strait, Chukchi Sea region, contact these people. Let us know what's happening. And just to emphasize that this is an issue to be worried about, I borrowed this slide from one of Gay's presentations. This is what I would almost call folklore. It's not clear when this happened, but in this region, Ipnarak, how do you say it? But anyway, it's near the Serpentine River. It's in the Seward Peninsula.
Ipnarak,
Ipnarak,
up by the Serpentine River, and it's not folklore. It's absolute traditional knowledge. So just
right.
semantics there. Thank you very much.
Yeah, wrong. I used the wrong word, but it's the kind of information we're looking for from the elders in various communities and others. Do you have records where people have gotten sick or died in the past, and where and what's the symptoms? And what this one says is that this was the location of a red tide at one time that caused many deaths. And it doesn't say what were they eating. I note that this is a place where there's a lot of fishing that goes on, including fishing for herring, one of those forage fish that I was talking about so was this herring that was eaten? Was it clams? Who knows? But notice this location. It's in that pathway of the transported blooms from the south. So long ago, there was that threat, and it got to the point where many people died. And that's entirely separate from this additional threat where we have this second mechanism of local cysts that are germinating. Okay, so to wrap this up, there are multiple toxic HAB species in the Arctic. I've only talked about this one, but there's a number of others that are likely to become more prevalent as things warm and as the sea ice reduces. We've documented 2 extensive seed beds of this toxic dinoflagellate, Alexandrium catenella. We've also shown that bottom temperatures are certainly high enough to support local germination. We've seen high concentrations of these vegetative swimming cells in surface waters, again, with temperatures supportive of cell division and increased growth and population development. And so taken together, this suggests that blooms in the region can originate and grow locally, in other words, north of the Bering Strait, and are likely to be self-seeding, means they're going to drop their own cysts up there and therefore recurrent. They're not going to be so episodic where they're dependent only on this transport from the south. But that's where evidence also suggests that southern populations are transported through the strait and are a second source of blooms north of the strait. So in a sense, it's a double whammy, as you might say. You've got 2 different mechanisms that can bring big populations into that region. And the situation that I've just described has very strong similarities with the Alexandrium bloom dynamics and distribution in the Gulf of Maine, where widespread blooms have persisted for decades. And it's been very useful for me to have the background of studying things so long in the Gulf of Maine and to see how those same tools and approaches and understanding are valid out here in Alaska. So the increasing risks of blooms also increase the risks of toxin in the food web, raising concerns for wildlife and human health. but we don't know how serious the risk is now or in the future. And that's in part due to the unique ecosystems and diet of the local communities. If everyone ate shellfish, if there was a huge shellfish resource out there in Alaska, we'd say, okay, this is going to be really serious. But we don't know what happens with seabirds or seals or walrus. And so we're trying to learn it, but we don't know what that risk is. So there's a multiple potential vectors or sources of toxicity, not only shellfish and crabs, but a variety of marine animals and mammals. But as I said, multiple projects are underway. We're trying to learn about this as fast and as much as we can. So my final point is that everything I've described here speaks to the potential for significant increasing HAB impacts on human and ecosystem health in that region. And just recognize, though, that many regions of the world, remember the map of the world with these red dots all over it, many regions of the world face similar risks and yet are able to maintain healthy communities and ecosystems. But it's done through good management, good communication, and through understanding what the threats are. So as a scientist, I feel like I'm walking a very narrow line where I'm telling you about something that's potentially alarming and worrisome, but I need to tell you that. I need to tell you and the world and publications and so forth that this is what's happening there. And then together we have to figure out how significant the risk is and how we can deal with it. To be honest, Kathy LaFay and I, as we talk about this, think that the human health risk is probably quite low, given again the diet and the ecosystems up there, and that the real threat may be one that's more food security and ecosystem health. You've already got many things going on in your region, that everything's warmer and the birds are starving and so many things are changing up there. And those ecosystems that are already stressed now have an additional stressor on them in the form of these toxins. And so that indirectly then can affect human health just through the availability of food and the security of food. And so that may be the real story here, not so much the direct risk to human health. So I just want to make sure I put that caution out there. There is a serious situation, but there's also a unique environment that we have to take into account. So I'll stop there. I want to make sure I acknowledge a number of different funding agencies and a lot of people who helped us collect samples and have been important in this study. So I'll stop there and hopefully people will have some questions.
Thank you, Don. I think people will have questions, but let's take a moment to catch our breath for all the news you just gave us. So thank you very much, Don. And I know people are really savvy with their clapping reactions, and people can do that. Also, Strait Science is pretty well known for being a grateful crowd because it's very late for you. Julie's giving you a clap right there, a video clap. So throw some love in for Don Anderson just for being able to present to us tonight. I know I have a lot of questions and some things I want to say, and I'm sure everyone else has. But let's make sure we don't lose our total perspective and thank our speaker for delivering the message. So thank you. You're getting a lot of
and thank you all. And don't worry about the time. This is important.
Okay, well, great. Well, I think we will probably have a number of questions. So with that.
Good evening.
Good evening. I'm going to go right to the callers first. Good evening, John.
Good evening.
Good evening. You have the floor.
My name is John Wally. I'm from St. Lawrence Island. I don't have any publications of your research. Would you give me an idea, or those of us from the island, what areas on St. Lawrence Island that you have done some research on?
Well, in terms of St. Lawrence itself, all the data I've been showing you has been from cruises that have passed by St. Lawrence and around it, but not on it. And Gay, maybe you can help here. We have a program going with about 8 local communities who are beginning to sample from shore. And I
Don?
don't know whether St. Lawrence is one of them.
May I interject, please?
Sure.
May I
ahead.
interject?
Go ahead, John.
Yes.
When we were at a meeting with the Alaska Eskimo Whaling Commission, there was discussion about this toxic substance, and they had been doing some research, and they were planning to do some research, and I recommended that they check north of St. Lawrence Island, because the two communities of Savunga and Gamble are probably the most unique user groups of marine mammals and seafood. Tunicates are very abundant, and the fall time we harvest them by beach combing, and both communities do harvest a lot of it. I see that you have been educated somewhat about our dietary traditions. We do eat the walrus stomach contents, and we had been told that there was some incidents of walrus even carrying this toxic substance. So, having said that, what are you professing to do to further make progress on the spread and this? Are you doing more research in the very near future?
Yes, there is some long-term funding, 3 to 4 to 5 years already in hand, and every sign is that this is likely to continue, because the work's been very productive in terms of scientific findings and publications, and it has generated a lot of interest. So yes, there is funding. There is this large project to look at food web transfer, and many of the organisms that you do mention as being potential foods are on our list to be sampled and analyzed, and tunicates aren't on there, and I'll have to pass that along to Kathy and see if we can start doing that. Gay, can you chime in?
I am going to chime in. So just because I know you're really involved in the algae, but there's a lot of moving parts, John. I'm really glad you tuned in and you have a question. So just for your knowledge, for the marine mammals and for testing around St. Lawrence Island, Don's mostly talking about the ships, and there's a lot of ships contributing samples. Most of them are these large research vessels, and they can't come in close just because it's too shallow by Savunga. They can't come in close. However, the AEWC is working, the Alaska Eskimo Whaling Commission is working with this project, and do know that every bowhead that is sampled from Koktovik to St. Lawrence Island, the feces and stomach contents, a portion of that now are delivered to Kathy LaFay, who's down with NOAA in Seattle, and she is on a plane right now, otherwise she would be on this call. And so for St. Lawrence Island and the Bering Strait region, from dead stranded animals, I will collect feces and stomach contents, whether they're seals, gray whales, beluga, and also the harvested animals are being sampled for AEWC. And there is also the walruses with the U.S. Fish and Wildlife Service doing the, I don't know, because I'm not involved in that, but the spring harvest collection that they do at Gamble and Savunga. They have also, some of the samples there are directly from the walruses. We also get walruses samples from Diomede, and I put in the chat, which I will send you these, I'll email these because I know you're on the phone, is that there was a walrus that was harvested at Diomede that did not act correctly. The guys called it into the beach, and it hauled completely out in front of people, and then did not put its head down and did not raise its head for another for a long period of time. It was dispatched, and the feces in that particular animal were 4 times over the seafood safety regulatory limit. That was in 2017. So Diomede was able to get do traditional estimations of the health of that animal. They were correct in their assessment, and they have brought very high quality information to this project. So I do want to give a shout out. There is information coming in from this region, the Bering Strait region, as well as the North Slope Borough. They are also from harvested animals and from dead stranded animals, if possible. There are levels being detected. But I have my own question, which I'll bring at the end, but I just wanted to answer that, John. I hope it helps.
Thank you very much. I would challenge you to go local and go to the tribes and work with them about the research. If you're going to actually localize it, I would challenge you to include St. Lawrence Island in your research, because it is definitely a hot spot. A lot of the Japanese current comes up, and then the prevailing wintertime conditions bring the ice pack down. In the late fall, because of the prevailing northerly, northeasterly winds, I would challenge you to go west to our closest neighbors and develop some kind of a relationship where you can do an international research in place. So anyway, thank you very much. I appreciate that.
I'd just like to say one thing with respect to your last comment. One of the fundings that we've recently gotten is joint with Russian colleagues. So if that's what you meant by your western neighbors, we already have that in mind. We will be getting samples from that western part of the Bering Sea, from the western part of the Chukchi Sea. We'll be working with them as well to understand the transport of these populations. One of the questions I see in the chat from Julie is, do you have an idea?
Before we go to the chat box.
Okay, sorry.
Before we go to the chat box, I know it's a normal thing to do, but people here are on the phone, and it is sometimes can be a very tenuous connection, and people have stayed with your talk the entire time. So I know it's a little bit unusual protocol, but out here we go for the phone first. Are there any other questions from the callers? And I appreciate each and every caller that's made it through this whole presentation. All right. Stay on the line, and if you want and you have any questions, I see there's a phone lighting up like someone's trying to talk. You may be muted, but not at this end. All right, we will just have to power through.
Rachel, I have a question.
Go ahead, Rachel.
Okay. The question is, you mentioned in the presentation, Don, that the seed beds were, I couldn't tell if it was the salinity was more of a determining factor where they're located, or was it more of the ocean current, or was it like both?
No, it's not. It's not the salinity. It's the currents. I didn't present that, but we have some nice data where we've got years and years and years of velocity data from cruises. Right, again, the very same region. If you started down near St. Lawrence and went all the way up, and all of these boats have been measuring currents as they go, and if you put that all together and take a look, what you find, it's not surprising that the water's really rocketing through the strait, moving very, very fast. But then as it gets into the Ledyard Bay area, it slows down a lot. And then there's a little bit more of a jet as you get up near Point Barrow on the west side of Point Barrow. And so the one cyst seedbed is right where you'd expect the water, since it's slowed down, you'd expect things to settle out easily. You know, we always if we have a high energy environment in terms of waves and currents, you don't get any mud or clay, you get sand. It's in the cysts act like the clay particles. So they will settle where it's calmer and deeper. So a combination of of bathymetry, the depth, and whether there's basins to collect this material, and the water velocities also then give you a seedbed. And the story, I don't exactly know the story over there by Utqiagvik, but one good explanation, not only does the water sort of slow down there a little bit to the east of the Point Barrow, but it's a bowhead whale hotspot. It's one that's been long known to be a feeding area for bowhead whales. And one of the reasons is it's got hydrography, it's got currents that concentrate the plankton food that they will have, in the fish and whatever else is always concentrated at these features. We call them fronts, just like fronts in meteorology. There's fronts in the ocean, and a lot of things happen at those fronts. And so I think that second seedbed may be related to a different part of the circulation, a different mechanism, but it still ends up with lots of cells accumulating there and then dropping cysts right below those features. So the answer, yes, is largely hydrography.
Thank you.
All right, great. Well, I'll just start chunking through the questions if you're up for it, Don. I know it's late,
Sure,
but
yeah, no
okay.
problem.
The first question was Franz Muter. Do shellfish, fish, seabirds, and mammals flush most of the toxins between one summer and the next, or do they accumulate through the years?
Well, the answer to that is both, but it depends on the species. Starting with shellfish, there are certain species that take up the toxin very, very quickly, but they also depurate or get rid of it very quickly. And that would be like mussels, would be at the very top of that list, because they filter so much water that we often call them sentinel species, because they're the first ones to get toxic, but they're also the first ones to lose the toxin. Other species, then there's this gradient that goes at the other extreme. You get the butter clam, for example. I know you've got butter clam on the west coast of the US. I don't know whether you have that same clam up in Alaska, but that will hold on to the toxin for years, long time. There's work ongoing with gooey ducks that maybe there's something there too, but so it really does depend. But certain species clean up quickly, others take much longer. And honestly, we don't know how to answer that question for fish and some of these other animals, because it just hasn't been studied, but needs to be. I think with fish, I can guess what the answer is. That think about where the toxin is in fish. It's in their stomachs because of what they've eaten. Then after they start to digest it, it goes into the kidney and the liver to sort of be detoxified or excreted. But the muscles don't accumulate any of this toxin. So if a fish is toxic and then it goes to an area that there is no bloom, my guess is that certainly the stomach contents are going to clear very quickly. What we don't know is how long the liver or the kidney will hang on to that toxin. But the good news is it's only a tiny part of the fish, and it's often a part of the fish that some people will just throw away.
And I know Julie Matthew, not to get into the finer points here, but she also studies that. And I think she's on the call as well. So if you want, you can message her if you're in the audience and have a more detailed question on that. You can go in the chat and instantly message Julie. Another question, unless I don't see any hands raised and people are being polite and not blurting it out, but if anyone has a burning question, we can alternate questions with the chat questions. Emma, go ahead.
I heard mention of livers in fish, and I'm just curious about that. Want a little more information if you can give it, because this is a time of the year known where people look forward to going out to fish for tomcod. And one of the best parts of the tomcod, other than the meat, is the liver. And a lot of times, you know, this fish is eaten frozen. Could you give a little more information about the livers in fish and something like tomcod?
Well, this is a situation where I'm going to recommend that you ask, or that Gay asks Steve Kibler to come and give a talk. He's been working on all these fish for a while, and I think he's got a really good story to tell and can answer that question better than I, because all I can do is tell you the things we don't know. I haven't seen the tomcod in his list of fish that he's been testing or learning about in terms of where the toxin is and what tissues. I also don't know what they eat, and you know where they might get toxin. I mean, that's part of the story. It's for these predatory fish, you know, what are they consuming, and where is that toxin? A walrus that digs clams from the bottom, that makes sense, but then I'm not sure what that would be. I'm not a fish person.
So to help you out there, Don, know that thank you. We will put Steve Kibler on the ropes, as well as Kathy, maybe, to answer some of these, and when we will focus, be able to have more conversations about this very eye-opening topic that you've sort of brought attention to. And for tomcod, Emma, I don't know if you're still on or you got bumped off, but know that if anyone has any concerns about tomcod, feel free to bring in a sample, give a call to me, 434-1149, and we will send that on to Kathy LaFay and get that tested. Just, you know, and I have a question, I'll wait till the end to give everyone else a chance, but I have a lot of questions about monitoring, who's in charge of HABs, and who do we need to talk to, those kind of things, but I'll get to that.
So what, maybe I just say one quick thing, because the other unknown, unfortunately, is we don't know how long, and it relates to the other question about how long this toxin may stay in these different tissues. So you were saying this is the time of the year when people start to eat tomcod. Well, this is also the time of the year when these blooms are pretty well gone. So it's a question of when the fish may have been exposed to the toxin a month or two ago, and how long they would hold on to it. That would be part of the answer, and I just can't give it.
And so because people have if you have any concerns about the food you're going to eat, let me have a little piece of it and we will send it on its way. It is research only, and it is extremely, well, I'll leave my little bit to the end, but we will try at our end the best we can to get what you have. You should not feel like you don't have any avenue to get your food tested. Right now, the only avenue is research. And so I will take that, and whatever you have of concern, whether it's a tunicate, a tomcod, a herring, a gray whale, whatever it is, I will try my darndest to get that run through. And I think there's a lot of people on this call that are willing to do that. And I think there are people on this call that are from further away in an urban-based center who have to talk to decision makers. Please let them know the state of what you're hearing. It is on these researchers right now to try to monitor, and I think that's extraordinary. But anyway, so Emma, go ahead. Did that help?
Yes, it did. I will definitely get you some frozen tomcod, Gay.
Awesome.
So we normally get them from out at Nook in the lagoon area.
Yep.
And what my concern is, if any of these that go into that lagoon have toxins that bioaccumulate in their system.
Well, there's a good way to at least get some information and hand it in, and someone will grind it up and get us an answer. I sure hope so.
Yeah, for sure.
So thank you, Emma, very much. All right, thanks. She gave a thumbs up. So another question is, do you have an idea where the southern populations, I guess that would be of your Alexandrium algae that got the toxin in it, that are transported through the Bering Strait originated, number one, and number two, where are the seed beds in the Bering Sea? Good
Okay.
questions.
Yes, they are good questions. The first one is one I can't answer with any confidence right now, but I can say that we're working on that. There's a number of different ways to look at it. Actually, let me do something here, if you'll bear with me, and actually share the screen again.
And remember, there's callers, so
Yes.
you can describe your slide. Thank you so much. I know it's late for you, Don, and you're doing great taking all these questions. And we very much appreciate it.
No, it's fine. I'm wide awake.
Okay.
So what I want to do is show this figure. And I said there's two different ways that we are going to be looking at this, what I would call a connectivity problem. One of them is a molecular, a sort of a genetic approach that we're going to be doing to try to take what we're starting with are cysts in a variety of locations that we're germinating, making cultures, analyzing their DNA, and doing something called microsatellite analysis, which allows us to try to draw connections between populations in different regions. And that's ongoing, but we have some preliminary data that I'm showing here. Now, I've been talking as if saxitoxin is a single chemical, and what it is a family of chemicals. There's more than 20 of them that just differ ever so slightly in their composition. And what happens is when an Alexandrium cell makes saxitoxins, it'll make like 3 or 4 or so of them. And a different strain from somewhere else will make 3 or 4, but they might be a different 3 or 4. And so we can use what we call that toxin composition profile as a fingerprint for these strains. And what this map shows, and again, for the people on the phone, it's a map that shows the Russian coast and then the Alaskan coast with these sort of pie diagrams on them. And the different colors are representing the different, we call them derivatives or congeners, the different forms of the saxitoxins that are produced by isolates from those locations. And if you start up north and look at the one at the very top in the Chukchi Sea, that one is almost red. And that is mostly saxitoxin, which is one of the most powerful of all of the toxins in that family. And if you look at all of the others around there, even way down to Kamchatka or down towards the Aleutians, there really aren't any that look like that one. So we're going to be doing this with 50 or 60 or 80 more isolates. We've already done the analysis. We just haven't figured it all out yet. But we will get more to compare. But that one looks like a bit of an anomaly. That one doesn't look like any of the ones to the south that we might point to. But if you look at the two on the far right that are from the Ledyard Bay area, and then you say, well, what do they look like? To me, I'm talking for those of you that can see on the screen, these two, to me, they're starting to look like some of these over here in Kamchatka and on the Russian side of the Bering Sea. Notice the ones we have over here in the east, and we don't have a lot. We've been trying to get sediment, trying to get more from this eastern Bering Sea region, but again, COVID's made it difficult. But the ones over here have a lot of yellow, and that's representing what we call C1 and C2 toxins, which are really weak toxins. They are like 1/100th as powerful as the saxitoxin or some of these others. And so this starts to tell us, I actually, if I had to bet, I would think that a lot of the cells we're seeing up north of the Bering Strait are coming from the Russian side over there, south of Chukotka and that area. There is a current that goes that way. They have found large numbers of cysts over in that eastern part of the Bering Sea. So there's a good chance that's the pathway that we will resolve with these two methods someday. But the final point is just that it's really important to know this, because if these yellow C1 and C2 toxins are the ones dominant in the organisms coming up through the strait, then that's much, much less of a health risk to animals or to people. Imagine if the toxin is 1/100th as potent as it is in a different isolate that comes over from the Western Bering Sea. So one of the things we need to know is what's coming your way, where is the source? And we're trying to understand that. And this is just one of the tools. And the final question for Julie, though, is because we are just struggling to get sediment from different cruises because of cancellations and so forth. We should be getting some sediment from the northern Bering Sea soon, but it's just something where we're dependent on other people to give us the samples. So I don't know where the cyst seed beds are in the Bering Sea. We've mapped out everything from St. Lawrence north, but everything from St. Lawrence south is very poorly understood right now.
All right, thank you so much, Don. And I'll go with the next chat question, which is from John Darwin. Thank you for your presentation. If there's, oh no, wait, I'm sorry, John. I got to go back up to Davis. Sorry, I'll get back to you. Davis with KNOM Radio asked, hello, Don, thank you for your presentation. You mentioned earlier low levels of saxitoxin found in fish, various marine organisms that are not shellfish. Compared to the FDA safe shellfish consumption levels, these are low. But is this still the best metric for scientists and researchers to use when comparing toxin levels, especially since we don't yet know if levels are high enough to cause health impacts in marine mammals?
No, it's a good question, because I mean, take the fish. I mean, the answer is no, I don't think it's the best metric at all. And think about what Gay just said about the walrus. I mean, you were measuring toxin in feces, all right? Well, what does that tell you about the toxin in the animal itself, or in its blubber, or in its liver, or its whatever? You know, it's not a good measure. Yes, it's above the limit for the mass of feces you have there, right? But it was per gram of feces. Am I right or wrong on that? Go ahead, Gay.
So for that question, know that there is, and again, this is the project with the marine mammals and the marine organisms. One of the things with that particular walrus that was harvested in 2017 that did have weird behavior, I immediately asked, was that enough to make a walrus that unaware? Everyone just kept saying he was so tired, he didn't even notice anybody. It was incredible that he crawled out, a bull crawled out of the water and basically laid down in front of people. So it was very, and they called him in. There were other walrus around with him, but it was so strange. So the answer I got was, we don't have a hot clue, because no one has ever looked at walrus and saxitoxin levels and what would even make a walrus woozy. Is it 5 times over the limit, or was that walrus have some other difficulty? So it became really apparent to me that there is a giant blank slate of information, and thus these researchers are trying to fill that in. One of the things with the walrus, that sampling program with the Fish and Wildlife Service at St. Lawrence Island, and what we ask for from anyone who's harvested and is sending in stuff, send in a piece of the intestine, send in some blubber, meat. There's really not a concern about meat and blubber. However, the intestine is that's where the toxin is transferred through. The stomach is where the poison gets squished into the bloodstream from. So let's look at that. And I believe Kathy LaFay is doing some of the first work. Again, this needs to be done 10 years ago, but I believe she is looking at intestine from the same animal as feces. We do that with the bowhead as well. We'll take a tiny little piece of the intestine as well as the feces, so that if it's hot, if it's a high level, can you look to see, is the intestine going to be something you shouldn't be consuming? Or, is there a chance of that? Or maybe after many, many animals get tested, no, it's not of concern. So there's much to learn, and I hope that helps answer that portion of what you were trying to get at, Don. Thanks.
Yeah, and Davis, I would just add that another example of how it's difficult is take the fish where the kidney and the liver are just a tiny part of this huge fish. If you took the amount of toxin in those tissues and divided it by the mass of the fish, you'd find the level to be extraordinarily low. Whereas if you just took the amount of toxin in liver and divided it by the mass of the liver, that can be high and dangerous. So it really depends. You have to be careful about that metric. But at least if people are eating livers or eating clams and so forth, then the metric is good, because it's that tissue that's actually being consumed. But you've just highlighted why some of these problems are so difficult for us, because a lot of this is new.
Hang in there. Thank you very much, Don. Rob Kaler with the U.S. Fish and Wildlife Service has his hand up. Go ahead, Rob. Good to see you on.
Hi, Don. I learned a ton. Thank you so much. Yeah, I know it's a little past midnight your time.
Don't worry. Don't worry.
Yeah.
This is fun. I'm enjoying this.
You're a trooper,
Science.
and thanks, Gay, for always hosting great meetings or great science talks.
Thank you.
I guess I'm wondering, that was a really cool, that curve that you guys have. I'll admit I did a screen capture, so I'm looking at it now, but the curve with the 85 heating degree days, is
Right.
is anybody, do you have any colleagues, perhaps Seth Danielson, kind of a predictive modeling? So if that type of, what you can look forward to in the Bering Strait and further north, Chukchi, in terms of that temperature range and what that might look predictively for.
Okay, so Seth is actually one of the co-authors on the paper we just published about this, but he provided more oceanographic data. But we are working with Bob Picard and his postdoc, Pagan, and they actually have taken data from that curve and applied it to those decadal temperature fields that I was showing, those 4 maps of the different temperatures over these 10-year intervals. And that allows you to, that's how we come up with that calculation of, over the last 20 years, the blooms would begin 20 days earlier. That's sort of averaged out using a model and using their physics over those whole entire seed beds. So we've already been doing what you're saying. What you need to do, what you're probably asking, is going forward, if we did some predictions about, let's say it warms another degree or 2, what can happen? And that hasn't been done, but all the models and the data are there in place to do that. And it would be we literally just did that analysis within the last 2 months. So looking ahead hasn't happened, but it can be and will be.
Excited. Thanks.
Okay.
We've got one more question in the chat. And before we do that, and then I have a question for you, but do any of the callers, because the callers have been steadfastly sticking with us, and I think that is, and I'm grateful, and we're not done yet, callers, but if you have any questions right now, you just feel free to speak your mind. So I know we're late and we're going back and forth with chat and people in the Zoom, but we haven't forgotten you at all on the phone.
Okay.
All right, thanks, Oprah. Are you you have any question?
Yeah.
Go ahead.
Yeah, I thought you were gonna one of the other chatters. Anyways, yeah, I'm Upik and I'm here in Diomede. I've been following along, and great presentation, by the way. I wasn't able to download the slides that Gay Sheffield sent me so that I could follow along, and I'm also using a landline to call. But so listening into your presentation, Don, I've noticed that you're mostly talking about Alexandrium, And so now I'm wondering, you know, after listening to everybody else's questions and comments about your topic today on harmful algal bloom, I'm wondering why you never focus so much on pseudonychia in your presentation, you know, and then listening to St. Lawrence Island and Gay Shepel talk about walruses and saxitoxins. I know that pseudonychia, I mean, domoic acid is a really important, is really important to study as well because it's produced by algae and it's produced by pseudonychia. So I'm wondering, you know, why pseudonychia wasn't studied so much in this presentation.
Great. Well, thank you for doing that. There's sort of two answers. The first is that you already see how much material I was trying to cover. I knew it was going to be long, and to add in a pseudonychia story would have made it even longer. But we are studying that, and I'm going to, you can't see this, but I'll describe this to you. I'm going to share the screen again. And this is a slide, just one. And again, I think that this might be another potential speaker, someone named like Kate Hubbard, who's doing all the pseudonychia work up there, might well be someone to have a talk here. You have a whole series of HAB talks, but I said there were two. I said there were two reasons why I didn't cover it. The second one, though, is that Kathy LaFay and I talk a lot about this, and we both feel that right now the biggest risk to the ecosystems and maybe to humans is from PSP right now. That the levels of domoic acid that she is finding in different animals, different tissues, are really very low, and it just seems that maybe it's a secondary worry right now. So that was the second reason. But just to give you a little feeling of what's going on, I've got a slide up now, and I'll describe it to you since you're on the phone. It's this big histogram on the bottom of all these bars with different colors, and the colors represent different species of pseudonychia. I mean, there's there's several dozen pseudonychia species, and some are toxic, some are not, some are strongly toxic, some are weakly toxic, and they're very difficult to tell apart with the microscope. In fact, you can't do it. You need an electron microscope for most of them. And so people have come up with genetic methods that can separate them. And this method that's described in this slide, it can't tell you, well, it just basically tells you of all the pseudonychia in a sample, how many are species A, how many are species B, how many are species C, and so forth. And what this diagram shows is that as you proceed from the west, like just right near Nome, let's say, and then you move along the coastline and eventually end up way over in the Beaufort Sea. That goes from left to right on the bottom of this diagram. And the height of the bars in a given color tells you the relative abundance of each species of pseudonychia that was observed at that location along the coast. And so in this left-hand part, which would be the Bering Strait and up to Point Barrow or Utqiagvik, that's sort of the first third of this diagram, you see a lot of dark blue bars here. And if you look down below, you know, that is a Pseudonychia delicatissima, which is toxic, though it's not hugely toxic. But the red that's right above it, and it's probably maybe anywhere from 20 to 30% of the pseudonychia that are present in these locations between, let's say, Nome and Utqiagvik. There's quite a few of these red bars at again, 10, 20% that are Pseudonychia australis. And that is the most toxic of all of the pseudonychia. And then there's some green bars that are another organism that's called pungens, Pseudonychia pungens. And these are weakly toxic also. So the story in the western part of this region is that there are toxic forms, including some that are very toxic, that produce domoic acid and cause, this amnesic shellfish poisoning or domoic acid poisoning in animals. And they're here, they're there, and they're, this plot doesn't say how many are there, but we have data on that as well. But what I find fascinating about this figure is also, if you look sort of at the rest of this, the last 2/3 or 3/4 of it, which means look from Point Barrow over to the Beaufort Sea, and you see all these, okay, great. You see all of these grays and blacks and other colors. And what those are Pseudonychia, but we don't know even what species they are. We know they're Pseudonychia, but we don't know what species. They are unknown to science. And so they could be toxic. They could be wildly toxic. They could be non-toxic. We don't know. But there's this huge potential for new species, new levels of toxicity, and just to be uncovered. But it's up in a region that's not well sampled, not well understood. But we do know at the very least that in that region that goes from Point Barrow down to Nome and St. Lawrence, you definitely have some toxic Pseudonychia and some that definitely cause serious problems in other parts of the country with their blooms. So, but that's like I said, it's another story. It's different ecology, different toxins, different everything, and worthy of its own separate talk.
Did that help, Opik? Hopefully, Don can send that slide and I can forward that on to you.
Okay, that would be great. Thank you. Yep,
Okay.
It kind of helped.
I know it's hard not to see it.
So
and-
I'm thinking, I never knew that there were so many other species of Pseudonychia. That would be another thing I would love to learn about. And then finding the other species of Pseudonychia and not knowing what they are, could it be from like the ocean being contaminated with all these other chemicals that they're, soaked up in?
Yeah, I've got two things to mention there. The first is that Jackie Grebmeyer is editing a special issue of a journal, and one of the chapters in it is going to be about Pseudonychia in this exact region. It's being authored by Kate Hubbard, and one person in my lab is a co-author. So, and it's very far along as a manuscript. So at some point there will be a paper talking about all the different species that are observed here and what they know. But then if your question about, it sounded like you were questioning about pollution or other issues that might be there. I don't consider that to be a factor at all in either of these HABs right now up in those waters. I mean, we haven't looked, to be honest with you, but the waters are so much cleaner than other parts of the world where we study these, and it may not seem that way to you, but it seems that way to me at least. And these are not species that we often see in polluted areas. The first Pseudonychia outbreak was up in Prince Edward Island in Canada, and people often talk about the pristine waters of PEI, but you can still have this very dangerous organism growing there. So I just think that these are ones that grow in natural waters and don't necessarily need any human or anthropogenic type of inputs to help them along.
Okay.
All right. Anything else, Opik?
No, nothing right now.
Thanks for your questions. Those were really great. And there's a question from John Darwin. Now, John is an archaeologist and he works in the Eastern Norton Sound and throughout the Norton Sound area. And he says, is this algae bloom growth a modern phenomenon? Or do you believe that historically this has happened on and off again throughout the Holocene? with periods when the waters were warmer?
Okay, well, that's a nice question also. In general, I mean, there's so many different algae causing blooms everywhere, but I'll start with Alexandrium. We have done some years ago some genetic studies where we were looking at isolates of Alexandrium from around the world, from the Atlantic, from the Pacific, and so forth. And what we found was that there were isolates from, let's say, Alaska or California that were considered the same species as what we were looking at on the Atlantic side, let's say in Massachusetts. But they were related, but they were different. And the best way to explain that was that at some point in the past, there was a common ancestor. And that goes back to the times when if you went up above North America, there were pine trees growing and it was warmer and it was temperate. And that there were, at that point, this Alexandrium, we believe, was up there already, and then it spread into the Pacific and into the Atlantic, but then it froze. It sealed up, and then they evolved separately. So you see, they had the same distant parentage, but that they then had plenty of time, millions of years, to evolve. So we believe that at least at the species level, they've been up in this region a long time ago. But what I think over the last, I don't know, 10,000 years, 5,000 years, they may not have been. It may have, again, have been too cold and the only inputs were coming in from the south. So one of the things we are trying to do in one of these cruises that's coming up is to take deep cores and to try to look and see how far down we can find the cysts of this organism. And even beyond when the cysts are going to survive themselves, look for their DNA. It's called ancient DNA that you can look for in these cores. So we're trying to address that question. But I do think that many millions of years ago, things were warmer up north in the Arctic, and Alexandrium was there then.
And I'd like to chime in if I can too, Don, if that's okay.
Sure, absolutely.
So John, your question on a much more recent scale, because you're talking Holocene, and I'm not, I don't know, that's
Right.
beyond my education. But, for me personally, there are outstanding historians with magnificent data sets throughout the region of the Bering Strait. And one of them was Winton Weapuk from Wales. And he's no longer on this earth or in this realm. And he was one who contacted me when the first information kind of came out about what is a harmful algal bloom and maybe we have this stuff. And he is the one that got me he knew there was something in the region that had historically been a problem regarding the red tide. And he, through a series of letters back and forth and communications, he got me to go to the Park Service and dig in a book. And that is the information that Don presented. That was actually thanks to Winton Weapuk Sr. And it really opened my eyes that one of the weakest points in this whole, and it's not the researchers, it's not a bad thing. They're looking at the ocean, the algae right now and trying to figure things out. But there's certainly a history of toxic algal blooms that has not been tapped, and that is by the historians throughout the Bering Strait region, in Chukotka as well as in Alaska. And there are several things that when you're talking with people from Chukotka, they'll mention potential fatalities that have happened, or people in this region might be more familiar with the quote stinky gray whale issue. That's where I can get into that later, but that's something more familiar in this region where gray whales are putting out a bad smell. And when you eat from them or try to eat from them. A lot of animals won't eat from them when they're fresh dead. You can get the tingling of the lips right away, and people know not to eat it anymore. So, and a gray whale is an animal that eats off the bottom. So I think that one of the things that really needs to expand greatly and quickly is this historical capture the knowledge that we hold in this region right now. And, you know, I don't know what it is because I haven't, but I think back honestly quite a bit for Mr. Wayapuk, and his reaching out with that story has led to something that is generating these questions. And so it's just one little piece of a great wealth of knowledge in this region.
And let me just emphasize that however we can do it, whether it's through personal visits, which we, again, COVID canceled them all, but we were going to be visiting communities. And one of my objectives was to talk to people and to the elders and ask them, do you ever hear, are there stories about this type of symptomology, or people dying or getting, you know, or even in some parts of the country, in the East Coast, they don't eat shellfish at certain times of the year. And I thought you told me once, Gay, that on the Chukotka side, there was when there's snow on the mountains, or when the snow, I mean, that's what we're looking for. And we'd love to hear from people, historians, elders, whatever, these kinds of stories that would help us understand the past.
Well, I think there needs to be a real concerted effort for that, because that is worthy.
Yes.
So thank you, Don.
Thank you for your answer.
Okay.
Thanks,
well,
John.
One thing is that the only reason I broke in is that I have animal subsets for the last 2,500 years, including shellfish. So maybe I'll go look, see if there's something I can do isotopically to see if we can see if those toxins are. It's just a thought. Thank you.
Awesome. All right. Anybody have a question? Any other question? Callers? High priority if you're a caller. Okay. I'm going to ask my question. Oh, go ahead, caller. I hear somebody. Oh. All right. So to educate us, Don, to me, this is an industrial strength problem. It looks huge and it looks frightening, honestly, to me, because I live here and I don't want to see anyone get hurt, and I don't want that ecosystem to be troubled. And we certainly have all that. Who is in charge of the algae? Is it NOAA? Who is the person or the entity? Is there, you know, our world, my world revolves around seabirds, marine mammals, fish. Those all have authorized federal agencies. It's like, I haven't found out yet. Who?
Yeah, it's the first thing
Who's
is
in charge of algae?
Well, nobody's in charge of algae. It's more that the NOAA has been given by Congress a lot of the funding and the authority to have a national program on algal blooms. And so they're funding not just this research, but a lot of other studies around the country. But then we're also getting funding from National Science Foundation. They care about this. But maybe the answer you're looking for is not so much the algae, but the toxins, because it's the state that decides on its regulations and how it's going to monitor and what it's going to do. It does that with guidance from the FDA, but the FDA doesn't have jurisdiction over the state shellfish. They can make recommendations on thresholds and things like that, but there's something called the Interstate Shellfish Sanitation Commission, ISSC, and it meets and comes up with regulations and testing procedures and all that, but individual states have to implement them. And so Alaska does that, but it's got such a difficult coastline to monitor that it basically says you should not eat shellfish in waters that aren't tested. And the only waters that are typically tested in the past were commercial operations where people had shellfish farms. Nowadays, you've got a number of tribes like the Sitka tribe and others that have developed and gotten money for instruments that can do testing, and they'll welcome samples from all over. So the number of areas tested is going up, but it is a state, typically, in my state, it's Massachusetts Division of Marine Fisheries. They decide what's safe and what's not.
Right. So I want to give a number. I want to say something about that safety issue. And I really appreciate you bringing this up, Don, because it's not an easy topic. Know that for the Bering Strait region, the Norton Sound Health Corporation, which is our hospital here, has had training specifically in 2019 for determining symptoms of both botulism, which is something that can happen here with devastating consequences, and also paralytic shellfish poisoning, which is poisoning by too much saxitoxin, what we've been talking about here. So the hospital here has been trained. Also, the clinicians have also been given a heads up. And for those who live here and know that oftentimes we are on our own, being so rural, that there is, by the state of Alaska, the section of Department of Health and Social Services, section of epidemiology, has a 1-800 line with 3 registered nurses 24 hours a day. And I want to give you that number because you don't normally see it. It's usually for given out to hospitals and clinics. So if they get a patient, they can call and chat and say, well, I think this person may, or what do you want us to do? That kind of thing. But in Alaska, I'm going to give that out because if you are in a rural community and you have a scare or you don't feel well or you're far from the clinic and have a cell phone, whatever your situation is, you should know these numbers. So I'm going to give these out if people have a pencil or pen. So get your pencil or pen. So 24/7, the state of Alaska has a number. Now there's actually 2 numbers because sometimes they're off hours, they're on the weekday, and then there's the people that are on the night shift. But there are always 3 registered nurses on call. The number is for Monday through Friday during the workday, something were to happen, 907-269-8000. If it's after hours, so if you can't get through on that 907-269-8000 number, you should call 1-800-478-0084. 1-800-478-0084. Someone will answer one of those numbers. That is what we've been told. And those numbers get called, and it's the real deal if you will have medical help on there. I also urge you to, the first line of defense is to get to your clinic or get a hold of your clinic so that they can call the Norton Sound Health Corporation, which is, of course, the best thing you could do locally. But if something is, you're not able to do that for whatever reason, those are the numbers to contact for help. And they will immediately start either giving you medical advice or getting other people to try to get help to you. So I just wanted to put that out. The topic is sort of scary to me, but there are answers. We live with it's nothing new in the Bering Strait region. We have lived and continue to live with low levels of saxitoxin. Our animals, we can see it, it's in the animals tested, but nothing catastrophic has happened. But we don't want to have a problem. So anyway, that's the number to call if you have eaten any kind of shellfish and are not feeling your best. Just like COVID, early testing, go call somebody and get some advice right away. I just wanted to add that in.
Good.
Mm-hmm. All right, and the rest I think is all kudos. Any other questions or comments? and again, I guess my other question is, Don, and then I'll stop, is, so if there's no one person in charge, how or who, if we have concerns, or we want to have more research, or we want to have more training, who then do we turn our, as American citizens, who do we turn to voice that concern and request for more help?
Well, actually, you can do a couple things there. One is that at a lower level, there are this NOAA program that I mentioned that is we actually have legislation from Congress for harmful algal bloom research, and it calls out NOAA to take a lead in all of that. It has interagency working groups, but NOAA is the lead. And within NOAA, there is one of the line offices called National Ocean Service, and within that is a program called NCOS, National Center for Coastal Ocean Science, NCCOS. And I know that and underneath that are the program managers that give out the money for various HAB programs. And I know that if a request goes in to NOAA and to, let's say, to NCOS or even just to NOAA, and it says, we need training, we need help up here in Alaska, that they will then make things happen. They listen to stakeholders. They listen to stakeholders a lot more than they listen to me and other scientists, believe it or not, because we're always saying, give me more money to study this. This is important. But if a bunch of people from a region say, or even several people say, this is important to us, they can make something happen. So that's one bit of advice. But another is to work with people like Steve Kibler or Kathy LaFay and others who work at NOAA at a lower level, but have a mandate to work on projects here. And they can help organize training courses or help make them happen. Or sample analysis or whatever. And I'm referring you to NOAA there, but you can also turn to people like me, and we have resources also that we can turn to. And one of the ones I can turn to is I go to Congress quite a bit. And I think what I could do is say, all right, Gay, get spiffied up and come down to DC, and we're going to go and visit some delegations and have an ask. An ask will be that Alaska needs this and Alaska needs that, and we'll help you do that. We've been doing that on a national level a lot, but it also needs to be done for each individual state so that Alaska can step in and support not only these national funding programs, but specific activities. So that's, believe it or not, they will listen at both levels that I described.
All right, well, I think we're going to have a lot of I appreciate that, Don. Maybe I'll talk to you a little bit more about that, but I think we should have many more straight sciences, and I wrote down quite a number of names here. So I'm hoping that we can all become more educated in this, this emerging significant issue that has real ramifications for human health, wildlife health, and food security. So I think we should have more of this until we get it figured out. Thank you very much.